Hi everyone. Welcome back. In today's video, we explore one of India's greatest scientific achievements, a breakthrough that could transform the future of genetic and hereditary diseases, especially for indigenous tribal populations worldwide who suffer from sickle cell anemia. Yes, this historic achievement is the development of BIRSA-101, India's first indigenous CRISPR-based gene therapy that may finally cure sickle cell disease. It is developed by Council of Scientific and Industrial Research CSIR, Institute of Genomics and Integrative Biology IGIB, and unveiled by Dr. Jitendra Singh, Union Minister Government of India. This breakthrough is not just scientific milestone. It is deeply cultural, emotional and historic. Before we proceed further, let us understand what is CRISPR technology. CRISPR is a powerful gene editing tool. It is like molecular scissors that can cut the faulty part of a gene, that is DNA, and help the cell repair it using the correct nucleotide sequence. In sickle cell disease, the problem comes from a single nucleotide mutation in the HBB gene at codon number 6 of the beta-globin chain. Here's what happens. Normal codon at position 6, GAG codes for glutamic acid. Mutated codon at position 6, GTG codes for valine. This occurs because the correct nucleotide, A, is replaced by the wrong nucleotide, T, in the DNA sequence of the beta-globin gene. This single nucleotide change replaces glutamic acid with valine at the 6th position of the beta-globin protein. This tiny substitution makes red blood cells hard, sticky, and sickle-shaped instead of smooth and oval. As a result, the body develops an anemic condition known as sickle cell disease. CRISPR fixes this by precisely removing the incorrect T and helping the cell restore the correct A, allowing the body to produce healthy, normal red blood cells again.
The Indian therapy BIRSA-101 uses an advanced CRISPR system known as an FN-Cas9,
an engineered version of the Cas9 enzyme derived from Francisella novicida. This specialized CRISPR tool is designed to edit the HBB gene with high precision and very low off-target effects. In this therapy, a patient's own hematopoietic stem cells are collected. The sickle mutation is corrected using FN-Cas9, and then the edited cells are reinfused back into the patient. Once these corrected stem cells start producing blood, they generate healthy red blood cells instead of sickle-shaped ones. Again, why is it named BIRSA-101? The therapy is named BIRSA-101 to honor Birsa Munda, the greatest tribal freedom fighter of India. He symbolized resistance, protection, and justice of tribal communities. Since sickle cell hits these tribal communities the hardest, the name is a tribute to their struggle and strength. In India, sickle cell disease is most common among tribal populations in Chhattisgarh, Jharkhand, Madhya Pradesh, Odisha, Maharashtra, Gujarat. Some tribal groups have 10–40% carrier rates, and many children suffer severe complications. BIRSA-101 was developed by CSIR and IGIB, one of India's top genomics institutes. It is being clinically developed with the Serum Institute of India, the world's largest vaccine manufacturer. This makes BIRSA-101 one of the first affordable CRISPR therapies in the world. The Indian government has announced a national mission, Sickle Cell Anemia-Free India by 2047. This includes large-scale screening, early diagnosis, and now, gene therapy-based cures like BIRSA-101. Sickle cell disease affects millions worldwide, especially in African nations like Nigeria, Ghana, DR Congo, and Uganda. Scientists and countries across the world are watching India's progress closely. Because if BIRSA-101 is successful and affordable, it could bring hope to millions far beyond India. India's BIRSA-101 is more than a scientific achievement. It's a promise to transform lives, uplift vulnerable communities, and lead global innovation. A new chapter has begun in the fight against sickle cell disease, and the world is watching. Thanks.